During the first meiotic division of mammalian oocytes, the meiotic spindle and chromosomes move from the interior to the periphery of the oocyte, so that the cell divides asymmetrically and extrudes a small polar body. Rong Li from the Stowers Institute for Medical Research in Kansas City, Missouri, is interested in how meiotic chromosomes make their move.
This migration process, unlike spindle positioning in mitotic cells, is not dependent on microtubules, but it's dependent on actin. If we get rid of the spindle microtubules, the chromatin can also move. So the question has been, how does actin really generate the force to move the spindle? One protein that's really important is formin-2, a formin family actin nucleating protein. Oocytes from formin knockout mouse cannot undergo this spindle migration process and fail asymmetric meiotic division.
One model proposes that myosin II pulls the meiotic spindle on a cytoplasmic network of actin filaments nucleated by cortical formin proteins. But another model suggests that formin II localizes around the spindle and generates a cloud of actin filaments that push the chromosomes to the oocyte cortex.
So in this particular study, we wanted to look at two things. The first thing is to really look at the spindle peripheral pool of formin II and actin and to get some better idea about whether they're really important for chromosome migration. And second question is really a question that neither model was able to explain, that is how symmetry breaking really occurs. How does the spindle end up moving to one side of the oocyte?
Li and colleagues, led by Kushi Yi, tagged formin II with an especially bright version of GFP to confirm that the protein accumulates around the first meiotic spindle of mouse oocytes, where it generates a cloud of actin filaments. But how does formin II localize around the spindle? Previous studies have shown that at this stage of oocyte maturation, the meiotic spindle is surrounded by both mitochondria and the endoplasmic reticulum.
So the next thing we did was to use markers for these two different organelles, and it turns out that formin II is associated with ER. And so that gave us some ability to disrupt this localization by preventing, for example, ER accumulation to the spindle periphery. And these manipulations prevented spindle migration, which gave us some confidence that this pool of formin II and actin are really important for spindle migration.
However, ER-associated formin II initially localizes all around the spindle, making it unclear how cell symmetry could be broken and the spindle moved to one side of the oocyte.
We thought that maybe one way to start to get some idea about this process is to do some careful life imaging and tracking of the chromosome movement. And then analysis of these trajectories immediately tells us that there are two processes. One process that seems very slow and not very directed. And then at some point when the chromosomes get to a certain proximity to the cortex, all of a sudden the movement becomes very fast and directed towards the cortex. So that tells us that there are likely to be two kinds of mechanisms that move the spindle.
Because chromosomes don't move at all in oocytes lacking formin II, the actin filaments nucleated by this protein around the spindle probably produce a pushing force to drive the first, slow and undirected phase of chromosome migration. But what happens in phase 2, when the chromosomes start to pick up speed?
At the same time, when the spindles start to move really fast towards the cortex, we start to see cytoplasmic streaming. The speed of cytoplasmic particle movement are the same as spindle movement. That suggests that the streaming accompanies the spindle movement.
During meiosis II, cytoplasmic streaming maintains the spindle and chromosomes near the cell cortex, so that upon fertilization, the second meiotic division also occurs asymmetrically to produce a second polar body and a mature ovum. Streaming is induced by a signal from the chromatin that activates the actin-nucleating ARP2/3 complex at the cell cortex, and Yi et al. Found that, as the spindle approaches the cortex in meiosis I, a patch of ARP2/3 starts to accumulate at the oocyte periphery.
And so we inhibit the ARP2/3 complex using this small molecule inhibitor and found that Now the spindle or chromosomes cannot undergo this second phase of very directed motion to the cortex. So really, our current model based on these observations is that the first phase really isn't a symmetry breaking phase. By having this actin polymerization at the spindle periphery, the spindle is basically experiencing a heterogeneous force. And this force is not directed, but it's enough to make the spindle drift in its position, basically destabilize this non-polarized state. Stochastically, when the chromatin wanders away from the center, if it reaches this threshold proximity to the cortex, it can activate the ARP2/3 complex. And then ARP3-driven actin polymerization generates this streaming and continue to push the spindle in that direction. And we also know that as the chromatin gets closer to the cortex, its ability to signal the cortex further enhances, and then that leads to more vigorous streaming. So this constitutes a positive feedback loop between chromatin-induced ARP2/3 activation and actin polymerization-driven streaming, which moves the chromosomes in spindle. So this is really what symmetry breaking occurs, making this movement in one direction irreversible.
Although formin 2 drives the first phase of chromosome migration by associating with ER structures around the spindle, it may also be involved in later stages of the process, as the meiotic spindles of formin 2 null oocytes fail to migrate even if they form near to the cortex where the chromatin signal should be able to activate the ARP2/3 complex and cytoplasmic streaming.
It seems like formin 2's function is not just to generate this initial movement. Maybe it is also important for some kind of communication between the chromosomes to the cortex. This signaling pathway is really not fully understood. So one could ask, how does formin 2 and ARP2/3 complex really communicate? And how is this chromatin signal conveyed to the cortex? And how does formin 2 actually localize to the ER?
And while Li and colleagues work on those questions, you can learn more about how sequential actin-based pushing forces drive meiosis I chromosome migration and symmetry breaking in oocytes in the paper by Yi et al., published in the March 4, 2013 issue of the Journal of Cell Biology.